All right. Thank you so much for the invitation. It's a really great honor to be a part of this NGL cohort. And I wanted to take this opportunity today to talk about what I see as some of the next generation challenges facing my field of statistical neuroscience and some of the ways that my lab is trying to start to address them. So my lab sits in the Wu-Tsai Neurosciences Institute at Stanford University. We sit in the Theory Center there, and it's a place where I'm really excited to be because it sits physically at the intersection of many of the things that I love. It sits at the intersection of neuroscience here in the med school, computer science, statistics, seats of machine learning. And so being a statistician, of course, I fit a line to these data points. In machine learning, we call this training a deep neural network. And I made a prediction, and that prediction is that I will also soon come to love geology. So stay tuned. It's also the intellectual center of a lot of the things that my group aims to do. It's at the center of data and interesting questions in neuroscience, the center of new modeling work and algorithmic work. And we really aim to be a bridge between these three things. So we spend a lot of time trying to think about how to develop new statistical models and probabilistic models for complex data sets, models that will help us to answer questions about high-dimensional neural recordings. Behind the scenes, we spend a lot of time trying to develop new algorithms that will allow us to fit these models efficiently and reliably. And then, of course, we spend a lot of time working with our experimental colleagues who sit right next door in this Wu-Tsai Neurosciences Institute to put these methods to use. And one of the reasons why I think this is such an exciting place to be working right now is because, as you all know, of the recent advances in neural recording capability that really offer great new opportunities to start to answer questions about neural computation. You're all familiar with two-photon and optical imaging techniques for studying large-scale neural population activity. Another thing that I'm really excited about is, on the behavioral side, getting more and more precise and rich descriptions of the motor outputs of the nervous system and the ability to collect these types of data side by side. So large-scale neural recordings in freely behaving animals performing complex tasks. Things like miniaturized fluorescence microscopes and the neuropixel probes, which we heard Nick talk really beautifully about earlier today. So I'm excited about taking these complex data sets and starting to think about how to build models around them. To give you a sense of the different types of data that I work with, I wanted to just run through a few of the different model organisms and types of ways that we've been thinking about them. So this character needs no introduction here at the Allen Institute. This is our favorite lab mouse. I started as a graduate student working with the data lab at Harvard Medical School to try to understand the behavioral repertoire of these animals and then relate them back to neural activity. And the way that we've been doing that is by putting top-down depth cameras overhead in order to measure their body posture over time. So here, this is on the right, this is the raw Connect video, a little bit of pre-processing. The top left is where we have found the mouse, rotate it, and aligned it so that it's always pointing to the right. And this becomes our behavioral video stream, which we think gives us a really nice description of what the animal is doing at any point in time. Increasingly, we can do this not just with the behavioral data, but as I was saying before, with optical microscopes. Here, a microendoscope that's going into the striatum and recording one photon calcium activity of about 100 or a couple hundred cells here. And so we can start to look at this behavioral data alongside the underlying neural activity and ask questions about the relationship between these two modes. This is going to be a common theme of the other examples as well. So this is, to give you another sense, this is a larval zebrafish, so a project that started, again, as a graduate student at Harvard, this time with the Engert lab. And this is a CGI rendering of the experimental setup. The fish is swimming around in a large tank, about a foot wide. And as it swims, we track it overhead with a high-resolution camera. And in this large tank, the fish explores a whole bunch more behaviors than it would when it's normally studied in neuroscience, head fixed in agar. And so we get out data sets that look something like this. Again, the fish is identified, tracked, and rotated to point to the right. And in every raw video frame shown on the left, we can go in and identify the eyes, draw circles around them, figure out where the fish is looking. We can find the tail and lay down a whole bunch of points along the tail. And we can go and see what the fish sees to some extent. We can find the paramecia in its field of view. So in this hunting sequence, you can see the fish execute a sequence of swim bouts. Its eyes converge as it finds a paramecia, tracks it down, executes a sequence of pursuit bouts, and then ultimately, here, captures its prey. And so we want to understand the sequence of different swimming behaviors, the types of behaviors, and the way in which those behaviors are executed and how that depends on what the fish sees on a moment-to-moment basis. Yeah, so the colors are there to tell you about eye convergence. So one of the things, if I play this again, you see most of the time it swims with its eyes out, but then when it enters into a hunt, its eyes converge, and that gives it higher resolution right in front of it where its prey are. And there's some interesting theories about why you might swim with your eyes out versus eyes open that would balance between keeping your eyes out for predators while at the same time having the focal resolution to track down your prey. And this is not work that I've been a part of, but I'm really excited about the potential to do the same sort of thing here, record neural activity in these freely swimming fish. This is a video from the Robson lab at the Roland Institute, where they're collecting, again, calcium activity, whole brain, in freely swimming larval zebrafish. So lots of opportunities to link up neural activity and natural behavior. The model organism that I'll talk about today is C. elegans. And C. elegans' behavioral repertoire is maybe not as complex as the other two, but nevertheless, it still goes between forward crawling, reversals, like you see here. And then every so often, as it crawls around, it will execute much deeper turns, which reorient it and point it in a different direction. And C. elegans obviously has a host of nice features associated with it. For one, it's only got 302 neurons. They all have a name. We know roughly where they're likely to be found. And we have genetic tools to do the same sort of thing, record optical activity. Sorry for the green bars here. Record neural activity here with nuclear localized calcium indicators in large fractions of those neurons. We have all sorts of connectomic information that we can eventually bring to bear for this model organism as well. So a really rich test bed for the types of models that we're developing. We can do this not just in individual worms, but for many worms of different genetic variants, different developmental stages, to really collect nice large-scale data sets of neural activity. But what I'd like to emphasize today is that data sets like these that I've been showing you really present a host of computational and statistical challenges that I and my group are trying to think about solving. And let me just lay out 3 of these that I think are really important and that are very pressing to the type of work that you all are doing here at the Allen Institute as well. The first is that these data sets are not well described by our traditional simple linear models. They're complex both in the spatial domain and in the temporal domain. And we need models that are able to capture this complex nonlinearity and relate them back to neural computation. The second, I think, is very relatable to you all here. In fact, I pulled this slide right from your website. These data sets aren't just simple single modality or single trial type data sets. These are heterogeneous data sets stitched together from many sources, collected over many individuals, many trials, many different brain areas and genetic variants. The list goes on. And so we really need to be thinking about how to develop tools that can synthesize these various types of data, put them together into a coherent modeling framework in order to make the most of these broad and diverse data sets. And then the obvious challenge is just that of dimensionality. I go around and I talk about how the labs I'm working with collect a terabyte of neural and behavioral data each day. I realize that here that number may not seem as impressive when I was just talking about 3 petabytes of connectomic data in the servers upstairs. But just to give you a sense, these are massive data sets. And if we really want to be making progress on these, we need algorithms that can iterate on these models at the scales that these data sets demand. And these are only 3 of many challenges, but I would argue that largely these break down into two types of needs. We need better models that can help us to capture these complex data sets and stitch together these heterogeneous data sources in a way that will reveal interpretable structure where and when it exists. And then we need algorithms that will allow us to fit these reliably and efficiently in order to test competing hypotheses. I won't talk at all about the algorithmic work that we do today. Instead, I'll focus on some of the modeling work, again in C. elegans, and try to go walk you through this process of how we start with really rich data sets like these and try to build models around them that will help to reveal structure in there underlying them. So once again, here is our video of neural activity in C. elegans, a couple hundred roughly 100 neurons here in the head ganglia. The very first step to this analysis is to take that video and line it up with a map like what I'm showing on the top here. This is from wormmaplist.org. So this map tells us where each individual cell is expected to be found, tells us what type it is, it tells us a whole bunch of information about these neurons. But of course, you can imagine that this alignment process is not particularly trivial, that figuring out which white dot corresponds to which neuron is not a straightforward mapping in all cases. And so what we end up with is for each individual worm, only a subset of all neurons can be reliably and confidently labeled. So here I'm showing you 2 minutes of data from the 5 worms in this data set. And in each worm, we range from anywhere from 30 to about 60 neurons, which can be reliably labeled. Nevertheless, we get a whole bunch of coverage, so we get many worms where we see overlapping neurons. Based on previous work from my collaborator Manuel Zimmer and Saul Cato when he was a postdoc in that group, we have some prior expectations for what types of things we might see in this data. And in particular, Saul in his work went and did, via a nice sequence of analyses, came to this hypothesis that the neural activity in this circuit actually lies on a much lower dimensional manifold. And that in this manifold, different types of behavior correspond to different parts of this trajectory on this manifold. And so this was a really exciting starting point for us, but some of the things that we were not so excited about or that we wanted to improve upon was the way that in previous work they went from that neural activity to this type of description involved a pipeline with with a sequence of temporal differencing and smoothing and dimensionality reduction and clustering, steps that all had associated hyperparameters and in the end seemed a little bit ad hoc. And so we wanted to come up with an alternative approach that might be able to generalize and to make more explicit what the assumptions were that were going into this model. And then secondly, in doing so, we wanted to have a way to take in these neural recordings from many different worms. Because right now, this pipeline is applied to sort of one worm at a time, and then you have to do some post hoc alignment to figure out how you take the manifold from one neural recording and map it to another. And the way that we're going to try to do this is by flipping this process around. And instead of thinking about how you go from data to manifold and segmentation thereon, we think about how you would start with a low-dimensional representation and then work up in a generative fashion to the neural activity. And once we've built that generative model, we'll invert it to reason about the underlying latent states and structure. So the first piece of this generative model is the assumption that underlying this neural recording is a sequence of discrete states. And intuitively, we're going to think of these discrete states as hopefully corresponding to the different types of behaviors that these worms execute. So maybe a discrete state for ventral turning versus forward crawling and reversals. And that that associated with each of these discrete states is a set of dynamics on this low-dimensional manifold. So here we can think of each of these dynamics as a function, a vector field, that tells you if you're at some point on this manifold and you're in this current state, i.e. this color here, where you'll go at the next time step. So once we have these two things, we can simulate forward or generate a sequence of continuous latent states, which you could think of as a trajectory on this manifold. And in this two-dimensional space, this corresponds to a two-dimensional time series unfolding over time. And so here you can kind of see in the yellow and the blue and the green states, you have these nice oscillatory dynamics, which corresponds to rotations in the space and oscillations in this time series. And of course, what we actually observe is not this low-dimensional state space. We observe some high-dimensional projection of it in the form of neural activity recorded over time. And what we're going to take advantage of is the fact that there are all these correlations and the wiggles across neurons that are really reflecting this underlying lower dimensional structure. So we're going to try to go from neural activity back to these continuous and discrete states and to their corresponding dynamics. One way that it's nice to conceptualize and represent these models is in terms of a graph structure. And these are probabilistic graphical models, and they tell us all the parameters that go into this model and all of the assumptions about how they relate to one another. So at the top, we'll have a big set of global parameters. This is going to correspond to all those vector fields, those dynamics functions. Once we know them, we can simulate this sequence of discrete states. Importantly, to get the next discrete state, you just have to know what state you're currently in. Once you have the discrete states, you can get your sequence of continuous states, and these are a little more complicated. You need to know not only where you are on this manifold, but what discrete state you're in, since that discrete state tells you which dynamics function, which vector field to apply. And then what we actually observe, the only nodes that are filled in this graph, are the sequence of neural activity, which are a projection of these continuous states. So this graphical model allows us to very quickly see the assumptions about what types of variables are there, how they're related to one another. What it doesn't tell us is anything about the parametric form of those dependencies. And so that's kind of specified separately. And to start with, we'll just assume the simplest set of assumptions. So we'll assume that the discrete states have some transition matrix, which tells you the probability of switching from the red state to the blue state, et cetera. We'll assume that the dynamics functions are all linear to begin with. So once you're in a discrete state, you just apply a simple linear function in order to update your continuous state from time t to time t plus 1. Linear functions are all represented by matrices and vectors, as shown here. And then we'll assume that the mapping from these continuous states to the observed neural activity is also linear. And these are all assumptions that we can start to relax and build upon in a very clear and explicit manner as we go forward. Under this set of assumptions, this is called a switching linear dynamical system, and it stitches together two things that we know and love: linear dynamical systems, i.e. Kalman filters, and hidden Markov models. And these types of models have been studied for many decades in statistics, machine learning, engineering, signal processing, but they don't necessarily capture some of the important features that we expect from that mental model that we started with. And importantly, one of the things that it's missing is this idea that different discrete states are going to be used in different parts of this manifold. So it doesn't say anything about the yellow state being used on the left, the red state in the lower right. And the solution to this is going to be to extend our model in a very simple way. We're going to add a new set of arrows that tell us that the next discrete state depends not only on what state you're in, discrete state you're in, but also where you are. And the way that you can think about these dependencies is that effectively they partition this manifold up into different regions with different dynamics. So you can have a blue state that's used in the upper left with one set of linear dynamics, a red in the upper right with a different set of dynamics. And by stitching together these linear dynamical systems, we can create a highly nonlinear system as a whole. So this is going to be a really powerful way to approximate nonlinear dynamical systems. The last piece of the puzzle is that when we go and fit this to one worm versus another, just little idiosyncrasies, perhaps differences in levels of the fluorescent protein expression, will give us slightly different dynamics, even if the worms are otherwise thought to be identical. So how can we build this model such that it's robust to slight variations in this underlying dynamical system? Well, the idea is again quite simple in terms of this graphical model. We'll postulate that for each individual worm, there's a switching linear system. We call these recurrent switching linear dynamical systems with these extra dependencies. And each worm will get its own parameters. That's going to allow it to have slightly different dynamics. But what ties it all together are a set of global parameters up top, which basically say that the dynamics and the parameters for each individual worm can't differ too much from the average. The other thing that this hierarchical model lets us do is to share information across worms, given that even though we only see a subset of all neurons. So if we only see a slice of the pie here, we can still share information via these global parameters in order to estimate not only the latent states and dynamics, but also the missing neural activity in other worms. So normally at this point, I would talk about how we're actually going to go and fit these models. As I said, we're going to skip that for today. If you're interested, please come talk to me afterward, because there's a lot of cool algorithmic work to be done there. But let me just jump straight to the end and tell you, what types of things we see when we fit these to this neural data. What we get out, first and foremost, is a set of, for each worm's neural activity, an underlying continuous set of states that summarize that high-dimensional neural activity, and a set of discrete states that segment it in time into instances of what this model has characterized as multiple instances of the same discrete state. And so here I've highlighted all the yellow states, and we see that they kind of correspond to similar patterns of neural activity across these worms, and similarly with the green states. We can look at this in a different way if we take just the first two dimensions of that continuous state and plot them as a trajectory in this two-dimensional space. And we can color code that trajectory by which discrete state it is in. As we expected, these discrete states are nicely localized in this space, and they all correspond to to corresponding dynamics functions, which tell you when you're in the discrete state that you go this way. When you're in the yellow state, you go this way, and so on. So it segments this time series up into pieces with linear dynamics that are sort of isolated in this state space. And then we can go back and ask, well, in each of these discrete states, what was going on in the neural activity? So we can say, in the blue state, these neurons were going up on average. The yellow state, these neurons were going down, and these neurons were going up. And since we know so much about what different neurons do in C. elegans, we can start to make hypotheses about what these changes in neural activity would correspond to behaviorally, even though in this particular data set, the worm is actually immobilized. And one of the nice things is we can go and take our inferred states and go back to the segmentations that Saul Kato found in their original paper, and we see a really nice correspondence between what we find with these recurrent state space models and what they found with their procedure. So that lets us assign some sort of behavioral interpretation to these discrete states. I motivated this model in the context of neural activity in C. elegans, but I want to make the point that the core modeling ideas have very broad applicability. And in particular, we can think about them as ways to segment high-dimensional time series data. Neural activity is only one type. Behavioral data, like what I've shown you here with this depth video, is another that I think is very much amenable to the same types of techniques. So here is applying those same sorts of models to this depth video of a mouse. The top is just a random projection of this video stream here. The middle here are the continuous latent states that we find. You can think of this as sort of the principal components or the eigenmice of this video frame. And the bottom are the discrete states which segment that continuous latent state space into different discrete syllables, as my collaborator Bob Data calls them. It's a little hard to see from this video what each of these syllables actually corresponds to. A better way to look at this is to take 50 instances of each of these discrete states, overlay them on top of each other, and time lock them to the entrance into that discrete state. So in these videos, which I'll play in a second here, what you'll see is that all of these time-locked instances of that discrete state will have a white dot that appears upon entrance into the discrete state, and then you'll be able to see consistent behavior across these instances. I'll play these a few times. So the top left one, you can see the mouse falling from a rear, synchronized with entrance into the state. The top right one, you see nice grooming behaviors. The bottom right one, you see one of the more common behaviors, which is try to escape the bucket. The mice never actually escape the bucket. It's kind of a sad truth of life. But nevertheless, this is a nice way of summarizing their behavior. And we can do these same things in the context of neural activity. We got to handle that cable that's coming out of the mouse's head, but we can do that with other techniques, computer vision, to basically identify it. And then we can say for each of these discrete syllables what was going on in, say, for example, in the striatum or the dorsolateral striatum in the direct and indirect pathways. So this should remind you very much of the types of things that Eric was talking about yesterday, giving us lenses into this behavior that we can then correlate with neural activity in these circuits of interest. The zebrafish is a little bit of a different example. And the reason is that the zebrafish behavior, if we watch this video again, it's not so continuous. The zebrafish swims in these punctate swim bouts, little brief flicks of the tail, in order to move through its environment. And so I think a better way to think about this, and what we've been doing, is to model this as actually a point process. So the zebrafish, each swim bout is a little discrete event. That event could have a type associated with it, pursuits versus prey capture versus turns to the left, et cetera, and maybe some continuous variability within those discrete bout types that we can model as continuous parts of the latent state. And the cool thing about this analysis is that we can go and try to predict what type of swim bout will come next, not just based on what it's been doing over past timescale or various timescales of its behavioral history, but also given what is going on in its environment. So we get things like these little Pac-Man style heat maps, which tell us probability of entering into hunts to the right, given that there is a paramecia in that location in the fish's field of view. So what comes next? In just the few minutes remaining here, I'd like to tell you a little bit about what we're doing now in the lab in order to try to build on these types of modeling techniques and address more interesting and more complex questions. And so one of the things that we recently have been working on that'll appear at NeurIPS this year is using these types of models in conjunction with deep neural networks and applying them to richer behavioral and neural recordings. So in particular, we've looked at some of the data from Nick Steinmetz's group, and which they very kindly made open source and is really awesome stuff. So here the behavioral videos are these zoomed in high resolution videos of the mouse's face. So you get a lot of whisker movement, you get nose pokes and things like that. And we've looked at this in conjunction with neural pixel recordings, in conjunction with wide field calcium imaging from Ann Churchland's group. And it's basically the same types of modeling ideas under the hood, but replacing some of those linear pieces with nonlinear pieces in order to capture these more complex dependencies. In light of what Eric told us yesterday about the relationship between activity in striatum and vigor, one of the things we're really interested in is how we can use these types of models to disentangle not only what type of behavior is being executed, but also how vigorously it's being executed. So we're building models that will allow us to say, this was an instance of a forward walk, but with this particular speed. And these are, again, they're going to look very similar in terms of models, but with an extra dimension of latent state, which is this underlying vigor variable. Much of the work that we've talked about has been building up the discrete transition structure, and it's been doing so in a fairly, data-driven way. One of the things I'm excited about is how we can bring in more top-down theory into these models, and what types of theories would make sense to bring into our models of behavior. Well, optimal control theory has a really long-standing tradition of being used in this field, and what we'd like to do is to infer not only what these animals are doing, but why they're doing it. And what might that answer look like? Well, it would come in the form of some sort of reward function for which this observed behavior and this observed sequence of actions is nearly optimal. We want to do this in a way that's more than just eigenmice or eigenworms. We want to do this in a way that respects the biomechanical constraints of this animal skeleton. And so this is a thing I've been talking to John a little bit about. Like, how do we start to take into account all of these complex dependencies that go from neural activity to ultimate motor production? We want to do this in the case of multiple agents and social behavior. And finally, totally different from all of these other examples, but I think really relevant to some of what you guys are doing here, is using these same types of models, which are, again, ultimately really good nonlinear function approximators in different domains, in particular for smoothing optical recordings of voltage. So voltage propagation within a dendrite is one of those things that has perhaps nonlinear dynamics within compartments, but we know a lot about the linear dynamics between compartments. So how can we build models that leverage that knowledge in order to build better models for this voltage propagation? In the spirit of open science, all of the methods that we're working on have corresponding code, of course, and that code we're working to make available in a way that's really easily usable. And I should say, first of all, this code that I've been working on for the past few years is what's under the hood in some of the IBL analyses right now. Tools for fitting hidden Markov models and all of their variations. And I would really encourage you to take a look if this is interesting to you. I'd really like to get some more feedback on what types of use cases would be best for us to focus on. So I'll end with this slide of what it is that my lab is trying to do here at Stanford as we get up and running off the ground. This goal of taking interesting neuroscientific data and questions, turning them into models and algorithms, and then putting them to use. There's a whole bunch of people to thank. I've had some amazing collaborators over the past few years. Certainly want to thank my funding sources and thank all of you for your attention. And I'll leave it here for any questions.
We have time for a few questions. Just want to raise your hand.
Great talk. Thank you. When you were looking at the model of the C. elegans, you showed how the model was useful if you have partially overlapping data to look at a hierarchical model. I was wondering, in the event that you have completely non-overlapping neural data, is it still useful to consider a joint population model like that? For example, if you had different cell types across individuals.
That is a fantastic question. Yeah. So we really leaned on that in the case of C. elegans, that we had these points where we could really make a strong correspondence between one data set and another. But I think that we can do something similar at a statistical level to bring together data sets where we don't have that luxury. And cell types might actually be the key to doing that. Can we say that we expect in various recordings from the same animal that there would be the same statistical structure of neural activity in cell type A across each of these instances? But I think the devil is in the details here, that we really want to be very explicit about what assumptions we're making as to what is shared and what is potentially different between individuals. So this correspondence problem that we've been talking about and that I saw on many posters yesterday, I think is getting to the heart of this, and I would really like to put that in a statistical framework that's very clear about here's how we think these different domains, these different recordings can be stitched together. Forrest.
Yeah, great talk. You touched at the end about this example where you were trying to factorize out speed from the behavior, but the brain is probably doing more than one thing at a time. So having just a single discrete state might not accurately capture all the distinct factors that are being kept update. Do you have any ideas about how you can fit models that would have multiple factors of state that would be more or less independent?
Yeah, that's an excellent question. Andrew Miri, I think, has been asking this question in regards to cell types as well. And I think that there's actually a really strong analogy to be made here. That in both of these cases, what we're doing is we're taking complex data, we're trying to boil it down into some space where we can cluster it. And then what can we use to explain variability within that cluster? And what happens when our clustering model is not sufficient to capture sort of odd, weird shapes in t-SNE space and has to split it into multiple spaces? I think this is fundamentally what's at the heart of this question. And accounting for speed by allowing speed to be basically a dimension that our behavioral states are invariant to is one way of trying to make a fairly explicit assumption about ways in which behavioral states in this case could vary but still be assigned the same discrete label. And you're absolutely right to think that there could be more than just speed at play. There could be a whole lot of internal state factors that are giving rise to that variation within this discrete state. And I can imagine what we're doing right now is trying to start with good ideas, good hypotheses about what those could be. But we're also thinking from a fairly generic standpoint of how can we just be robust to variation without a priori having to specify all the types of variation that may exist there, using ideas from robust statistics. Eric.
Thank you, that was beautiful. I wonder if you have thoughts you could share with us about how the connectivity matrices among the discrete states, the A in your work, or perhaps those A projected up into the higher dimensional space of neural activity patterns, relate to, in any way, connectivity among the cells themselves, or could be constrained by info about that connectivity.
Yeah. So in C. elegans, for example, this is a place where it's kind of the obvious next step for us, because what we've done is kind of describe dynamics at this level of abstract, continuous latent states. But of course, this is a place where we know a lot about physical connectivity between individual neurons. And so I think this question is really one of how do we relate technical jargon here, how do we relate autoregressive models defined at the level of neuron-to-neuron interactions to these low-dimensional state space models defined in terms of dynamics in some low-dimensional space? And so one of the things that I've been really excited about, I don't have much to tell you right now in terms of progress on this, but I think that of all the many realizations of neuron-to-neuron interaction models that could give rise to the same dynamics, only a few will actually be consistent with the known structural connectivity. And there's some really great work that came out of, I think it's Cook et al. This year in Nature, just kind of going back and revisiting this connectome of C. elegans with much more fidelity in both sexes to give us a lot of structural constraints for pinning down how these dynamics are realized in terms of, say, synaptic weights between neurons.
So I just wanted to understand, so the global parameters that you have in the hierarchical models, do you have any physical significance you can relate it to them? And what are the dimensions of those parameters? And also one more question is about when you have animal-wise like one more hierarchy added to that global parameter, which is different for each animal. Was there significantly different in those parameters for different animals?
I think the first question is very much related to Eric's question from before of how do we provide some mechanistic interpretation of these dynamics matrices. So the global parameters, sorry I was a little bit vague on this, the global parameters basically correspond to the A matrices in the linear dynamical systems and to the transition functions which partition this space up. And in that case that I showed you from the Kato et al. 2015 data set, there's not really a whole lot of interesting worm-to-worm variability. We don't expect there to be either because these should, for all intents and purposes, be basically identical worms. But the places where we do see interesting variability are when we apply this to a data set which I didn't show you, which is from a more recent paper from Manuel Zimmer's group, where they have recordings from many worms of different genetic variant and of different developmental stage. And there, the types of things we see are that worms at certain developmental stages and genetic variants will respond more or less to oxygen concentration in their tendency to execute forward-crawling types of behavior. So these types of hierarchical models are not just useful for getting some robustness to individual variability, they can also give you a way to characterize more interesting differences between groups that you think should exist or may exist. The second question, sorry, I spent a long time answering the first question. Now I got to remember what the second question was. Could you remind me? Yeah, okay, maybe I did answer. Okay, yeah, the variability. Great, thank you.
So I have a slant on Eric's question, which is in terms of interpreting the continuous latent variables. And it seems like, in principle, if you could measure every neuron's activity, you might not need that.
Yeah.
But I could also imagine how it could be useful for the modeling interpretation, whether it's neuropeptides or something. So if you had some thoughts on mechanistic interpretation of those.
That's a good point. So the low-dimensional continuous states kind of play two roles. One is that they allow us to sidestep a question of how you could learn a neuron-to-neuron model given only partial recordings, because it's not entirely clear how you do that. How do you fill in the parts that you don't see? State space models can give you a way to do that by assuming conditional independence given this latent continuous state. The other thing that they do is they play a form of regularization. So in terms of the number of parameters that these models have to fit, it's basically quadratic in the dimension of the continuous state and linear in the number of neurons. Whereas the number of parameters in this neuron-to-neuron model could, hypothetically, it's going to scale with the number of non-zeros in your adjacency matrix, which for C. elegans is going to be roughly 7,000 synapses. So not incomprehensible that we could fit that, especially as we start to record longer and longer duration recordings. But as a first pass, these models have a much smaller number of parameters.
Okay, let's thank Scott again for an awesome talk.